Welcome to Lab 3. In this lab, we start with cell cycle, and then later on, it will be the types of tissues. Look at this cycle. Numerous cells go through the cell cycle, which is an ordered series of events from cell growth to cell division or replication. And there are four parts, four phases of cell cycle. G1 phase, that's the first growth phase, in which the cell grows, develops, and carries out events that are unique to that cell type. Some cells, they never divide. This is the mitotic stage and remains in the G1 indefinitely. These cells are said to be in the GO phase or G naught, that's the word, G naught phase of cell cycle. Stage 2, the S phase or synthesis phase, where the DNA in the cell's nucleus is replicated. Then G2 phase, second growth phase. Throughout this phase, the cells perform activities to prepare for cell division. And then comes the most important part, is the mitosis phase or M phase. In this period, the cell cycle during which the cell, sometimes called the mother cell, will undergo the process of mitosis. And through mitosis, replicated DNA is equally divided between two separated nuclei. In most cells, another event called cytokinesis also occurs during M phase. Cytokinesis involves the division of mother cell's cytoplasm, which is distributed between two daughter cells. They are genetically and structurally identical to the original mother cells. The period in this cell cycle from G1 through G2, this part is known as interphase. Right? When the cell is in G1 phase, it has only one single copy of DNA. When in S phase, DNA replication occurs. In human, we have 23 pairs of homologous chromosomes. One chromosome of the pair comes from mother, other from father. After the DNA is replicated, each homologous chromosome exists as a set of identical chromosomes called sister chromatids, which are joined together as a region called centromere. So let's go to the mitosis. Mitosis has these steps, these sub-phases. The first phase of mitosis is prophase. In prophase, chromosomes condense and becomes visible. Spindle fibers will emerge from the centromeres, centrosomes, nuclear envelope breakdown, and centrosomes move toward the opposite poles. This is what happens during the first phase, prophase. Next, the pro-metaphase. Chromosomes will continue to condense. Kinetochores appear at the centromeres and mitotic spindle microtubules attached to the kinetochores. Stage 3, metaphase. That's interesting because you see those chromosomes are lined up at the metaphase plate. So metaphase, they come at the middle. This is how you can remember. Each sister chromatid is attached to a spindle fiber originating from opposite pole. In anaphase, they are apart. They are going in another direction. Centromeres is split into sister chromatids. Now they are called chromosomes. They are pulled towards opposite poles. In telophase, chromosomes arrive at opposite poles and begin to decondense. Nuclear envelope will start to appear. The mitotic spindle breaks down and spindle fibers continue to push poles apart. So up to this point, this is what happening during the mitosis, the cell part, nucleus part. But the last one is the cytokinesis. This time in animal cell, there will be a cleavage at the middle. That furrow separates the daughter cells. In plant cells, a cell plate, the precursor to a new cell wall, separates these daughter cells. So those are the things. Once again, prophase, they start to the preparation for the first phase. These are the things that happen before the nucleus is divided. Pro-metaphase then goes to metaphase. They come to the middle. Anaphase, they are broken apart. The pair is broken and moving to the opposite direction. Telophase, two chrome 2 nuclei are formed. And then lastly, the cell is divided in cytokinesis. In microscope, under microscope, we can find those things and they are visible. And you can try to correlate the features to identify what is happening in that stage. So this is important for everyone to know what are those different stages of mitosis and how to identify them. Like you see here on the left upper corner, interphase, nothing is happening. The cells are actually in that phase from G1 through G2. Second picture is the prophase, when the nuclear envelope is broken, chromosomes are condensed. When they come at the midline, that's the metaphase. They are broken apart, anaphase. And then telophase, two separate nuclei are formed. And there is the cell is already broken here in this cytokinesis part. I mean, they are divided into two parts, right? So that will be interesting for you to observe under microscope. In the second part of this lab 3, we will be learning about tissues. First thing, what is tissues? A group of 2 or more cell types that work together to perform common functions. And there are 4 basic types of tissues: epithelial, connective, muscle, and nervous tissues. Would like to start with epithelial tissues. As you see in this slide, there is a single layer of cell, flat cells. So whenever there are single layers, we use the word simple epithelium. And epithelium is the covering cell. And if the cells are flat, we call it squamous. So simple squamous epithelium, that means single layer flat cell. They have centrally located flattened nucleus. You see here in this top layer of this tissue, that's the simple squamous epithelium. In the second slide, we have simple cuboidal. This time, yes, simple is the single layer, but you see the cell layer covering the hollow. This is not the flat shaped, they are rather cuboid shaped, cube-like. So this will be simple cuboidal epithelium, one layer of square or cube cell. They have the spherical central nucleus inside the cell. In third type of cell or epithelium, we see single layer, but the cells are columnar. All right, so they tall, single layer of tall rectangular cell. This is called simple columnar epithelium. Again, simple means single layer, and according to their shape and size, we name them either, if it is flat, we say squamous. If it is square shaped, that is the cuboidal. If it is rectangular, that will be columnar. Some of them have cilia and folded into microvilli. You see in the picture on the right side, that upper part, some of the cells are containing cilia, hair-like structures. Yes, we find the cilia here as well. But this time, This is interesting because even though there are one layer of cell, but looks like multiple layer. If you see clearly, at first glance, it looks like multiple layer, but actually one layer. This is why it is called pseudo-stratified. Pseudo means fake. Stratified means multiple layer. So fake multiple layer cell, and they are columnar because they are tall cell, rectangular cell. So all these four types of cells that we discussed so far, they belong to the simple epithelium. Again, if it is flat, simple squamous. If it is a square, simple cuboidal. If they are rectangular, simple columnar. And if they look like multiple layer but actually one layer, this is pseudo-stratified columnar epithelium. These are all single layer. What if there are multiple layers? Like you see here, many of those cells are here, not one, two, or three. Many of those layers are here. So we use the word stratified. Stratified, multiple layers. If they are flat, we call it multiple stratified squamous. Like in this case, they are stratified squamous. At the same time, they have these cells are non-keratinized. There is no keratin. It is usually found in vaginal epithelium. You can also see the basement membrane here. Under epithelium, we'll be able to find this non-membranous layer, basement membrane. Just to go back a few slides, let me see. 1 second, almost there. So I was just going to show you basement membrane here under epithelium. There's a very thin layer, which is non-cellular layer. Right? Just to show you another basement membrane here, that pink color line just underneath the layer of simple or any kind of epithelium. This is also stratified because more than one layer, but this is cuboidal because they are almost a square shaped. Stratified cuboidal epithelium. This is found in sweat glands, the duct after sweat glands. This type of epithelium can be found. Next one is the stratified columnar. So more than one. Stratified means more than one layer. If they are rectangular shaped, tall, we call it columnar. And this type of cells are found in salivary gland ducts. This one is interesting. The bladder, inside the bladder, we find this multiple layer epithelium. That means it will be stratified. But they look like cuboidal here, but when the bladder is stretched, they can become flattened. So they change their structure, they change their shape. So we call it transitional. Transitional epithelium in the urinary bladder, they change their shape. When the bladder is empty, they will look like this, almost cuboidal, but can become flattened when this is stretched. Okay, so these four types of stratified epithelium, that means they have multiple layers of epithelium, and it can be stratified squamous, non-keratinized, found in vaginal wall, keratinized, found on skin, then stratified cuboidal, the ducts of the salivary glands, stratified columnar, and transitional epithelium. After the epithelial tissues, the next type of tissue is the connective tissue. And we find this connective tissue here, which is the loose connective tissue from skin. Usually just under the epithelium, this type of connective tissues are found. You see those cells like fibroblast, elastic fibers, collagen fibers, and and they are very loosely connected. Just under the skin epithelium, this type of loose connective tissues can be found. The opposite of loose will be dense. Dense, irregular connective tissues that is found in thick skin. You see, they are very dense in comparison to the previous one. That was the loose. This one is dense, and we see they have the ground substances, fibroblast, but they are irregular collagen. That's the point, irregular. If that was dense irregular, now the dense regular one. In our tendons, we find these dense collagen tissues, but they are arranged in a regular fashion. Right? You see those cells, the tissues, ground substances, collagen bundles, they are placed in dense, regular, collagenous connective tissue form found in tendons. Dense, regular, elastic tissues. In those organs which needs to do some work and need the property of elasticity. Think about heart muscle, heart tissues. They have to expand and then constrict, right? Contraction, dilation. Those things are needed for these organs. So the dense, regular, elastic connective tissues are found in those blood vessels, especially the big one, like aorta, close to the heart. Those or wall contains parallel elastic fibers. After that, we have the reticular tissue from the lymph nodes. And these are the reticular fibers. You see that they have leukocyte, white blood cells. This is taken from the lymph node, which is a part of lymphatic system. The reticular means net-like connective tissues. We are discussing different types of connective tissues. The one was epithelium first, then this one is the connective tissue. One of the connective tissue contains fat cells. Adipose tissue is that. From adipose tissues, they have those fat cells. You see that white bubbles, and these cells are called adipocytes because most of the cell contains this cell adipocytes that contains the fat with lipid inclusions. They store fat. So these are the different types of connective tissue proper. I mean, it can be loose connective tissue, it can be dense. Within the dense, we have irregular, regular collagenous, regular elastic, and then reticular connective tissues and adipose connective tissues. We may have to identify these pictures or different types of slides like this in our exam. Special connective tissues are bone and blood. As you see here, this is the bone, actually cartilage. The hyaline cartilage in our trachea, we find this type of cells, chondrocyte. Chondro means cartilage. So cartilage cells are the chondrocyte. It looks like this under microscope. Fibrocartilage, that's another type of cartilage. Then we have elastic cartilage from the epiglottis. So these are different types of cartilaginous tissues. But the bone tissue is pretty interesting for you because at first glance, it looks like a black hole. At the middle, that central canal, which contains the blood vessels. But you see surrounding that central canal, those black spots, looks like some insects, osteocyte, bone cell. Oste is bone, cyte is cell, so bone cell is osteocyte. So this is bone tissues. Another special connective tissue is blood, and earlier I showed you a blood cell under microscope. Again, most of the cells without nucleus, you see them reddish color. Those are the red blood cells. The one with purple color, these are the white blood cells with deep, uh, deep color, deep stained. Those things you see the nuclei inside the white blood cell. So these are different types of connective tissues. Our third type of tissue will be muscle tissues, muscular tissues, and this is the skeletal muscle where we see those striations. Skeletal muscle cells, they are multinuclei. I mean, multiple nucleuses will be there with the striations. This is how we can detect, we can diagnose skeletal muscle. Cardiac muscle, its important feature is that we will have intercalated disc. You see the dark line. These cells are branched cells connected to each other so that they can work together. And they also are striations. So two types of striated muscle, cardiac one and skeletal muscle. But this one must have intercalated disc and branching. Third type of muscle is the smooth muscle, no striation, no intercalated disc, and these cells are having only one nucleus inside, not multinuclear. This is pretty smooth, and this is called the third muscle, smooth muscle. Our last and final type of epithelium will be nervous tissues. And what do we see? There are many things in the nervous tissues, but most important character will be the neuron. Nerve cell, neuron, which contains nucleus. Nucleus you see here, cell body, then dendrites, axons. This is an interesting cell, neuron. We find in brain, spinal cord, in our peripheral nervous system. Right? So these will be the things we need to identify in our lab 3. Thank you very much.